Hello friends, today we'll learn about microbial production of organic acids. So let's begin. The major organic acids of commercial importance produced by fermentation are citric acid, gluconic acid, lactic acid, acetic acid, ascorbic acid and itaconic acid. These compounds may be produced directly from glucose, example gluconic acid, or formed as end products from pyruvate or ethanol, example lactic acid and acetic acid. The production of most of the organic acids is directly or indirectly linked with Krebs cycle. Besides microbiological production by fermentation, some organic acids are also produced by chemical methods. Example acetic acid and lactic acid. Now let's start with citric acid. Citric acid was discovered as a constituent of lemon. Today we use citric acid as an intermediate of ubiquitous Krebs cycle, that is citric acid cycle. We can say it as citric acid cycle, and therefore it is present in every living organism. In the early days, citric acid was isolated from lemon that contains 7 to 9% citric acid, and today about 99% of the world's citric acid comes from microbial fermentation. Now let's know about application of citric acids. Citric acid, due to its pleasant taste and palatability, is used as a flavoring agent in foods and beverages. Example jams, jellies, candies, desserts, frozen fruits, soft drinks, wine, etc. Besides brightening the color, citric acid acts as an antioxidant and preserves the flavors of foods. It is used in the chemical industry as an anti-foam agent and for the treatment of textiles. In metal industry, pure metals are complexed with citrate and produced as metal citrates. In pharmaceutical industry as trisodium citrate, it is used as a blood preservative. Citric acid is also used for preservation of ointments and cosmetic preparations. As iron citrate, it serves as a good source of iron. Citric acid can be utilized as an agent for stabilization of fats, oils, or ascorbic acid. It forms a complex with metal ions, that is iron, copper, and prevents metal catalyzed reactions. Citric acid is also used as a stabilizer of emulsions in the preparation of cheese. In detergent or cleaning industry, citric acid has slowly replaced polyphosphates. Now let's know about microbial stains for citric acid production. Many microorganisms can produce citric acid. The fungus Aspergillus niger is also commonly used for industrial production of the citric acid. The other organisms include A. clavatus, A. venti, Penicillium luteum, Candida catenula, C. Guillermondi, and Corynebacterium species. For improved industrial production of citric acid, mutant strains of A. niger have been developed. The strains that can tolerate high sugar concentration and low pH with reduced synthesis of undesirable byproducts, that is oxalic acid, isocitric acid and gluconic acid, are industrially important. Now let's know about microbial biosynthesis of citric acid. Citric acid is a primary metabolic product of primary metabolism formed in the tricarboxylic acid, that is Krebs cycle. We can say it as Krebs cycle. Glucose is a predominant carbon source for citric acid production. The biosynthesis pathway for citric acid production involves glycolysis wherein glucose is converted to two molecules of pyruvate. Pyruvate in turn forms acetyl coenzyme A and oxaloacetate which condense to finally give citrate. The major steps of the biosynthesis of citric acid we will now discuss. Now let's know about the enzymatic regulation of citric acid production. During the synthesis of citric acid, there is a tenfold increase in the activity of the enzyme citrate synthase. That is aconitase, isocitrate dehydrogenase that degrade citric acid are reduced. However, recent evidence does not support the theory and that reduction in the operation of tricarboxylic acid, that is degradation of citric acid, contributes to accumulation of citric acid. Increased citric acid is more likely due to enhanced biosynthesis rather than inhibited degradation. Further, there are anapleuritic reaction that replenish the TCA cycle. TCA means tricarboxylic acid cycle intermediates to keep the cycle continuously in the operation. Now let's know about pyruvate carboxylase. Pyruvate carboxylase that converts pyruvate to oxaloacetate is also a key enzyme in citric acid production. Now let's know about yield of citric acid. Theoretically, the yield of citric acid for the most commonly used substrate, sucrose, has been calculated. It is worked out that from 100 gram sucrose, 112 gram of anhydrous citric acid or 123 gram of citric acid to 1 hydrate can be formed. However, due to oxidation of sugar to carbon dioxide during triphosphate, the yield of citric acid is lower than the calculated. Now let's know about factors in the regulation of citric acid production. Strict maintenance of controlled nutrient conditions is very crucial for maximal production of citric acid. The optimal conditions that have been worked out for a nicer for the production of citric acid now we'll discuss. Now let's know about carbohydrate source. A wide range of raw materials can be used for the supply of carbohydrates. These include molasses, that means sugar cane or sugar beet, starch from potatoes, date syrup, cotton waste, banana extract, sweet potato pulp, brewery waste and pineapple waste water. A high yield of citric acid production occurs if the sugars that are rapidly metabolized are used, example sucrose, glucose, maltose. At present, cane molasses and beet molasses are commonly used. The variations in the composition of molasses, that may be seasonal and production level, have to be carefully considered for optimizing citric acid production. The concentration of carbohydrate significantly influences citric acid production. Ideally, the sugar concentration should be 12 to 25%. At a concentration less than 5% sucrose, citric acid formation is negligible and increases as the concentration is raised to 10% and then stabilizes. It is believed that a high sugar concentration induces increased glucose uptake and consequently enhances citric acid production. Now let's know about trace metals. Certain trace elements, iron, copper, zinc, manganese, Mg, copper, are essential for the growth of a nicer. Some of the trace metals, particularly Mn2 plus, Fe3 plus and Zn2 plus, increase the yield of citric acid. The effect of manganese ions has been investigated to some extent. These ions promote glycolysis and reduce respiration Both these processes promote citric acid production. As regards iron, it is cofactor for the enzyme aconidides. It is estimated that an Fe concentration of 0.5 to 0.05 to 0.5 ppm is ideal for optimal citric acid production. At higher Fe means iron concentration, the yield is lower, which can be reversed to some extent by adding copper. Now let's know about the pH. pH of the medium influences the yield of the citric acid, and it is maximal when pH is below 2.5. At this pH, the production of oxalic acid and gluconic acid is suppressed. Further, at low pH, transport of citric acid is much higher. If the pH is above 4, gluconic and accumulates at the expense of citric acid, and when pH goes beyond 6, oxalic acid accumulates. Another advantage with low pH is that the risk of contamination is very minimal, since many organisms cannot grow at this pH. Now let's know about dissolved oxygen, dissolved O2. The yield of citric acid production substantially increases when the dissolved oxygen tension is higher. This can be achieved by strong aeration or by spurging with pure oxygen. It has been observed that sudden interruptions in oxygen supply cause drastic reduction in citric acid production without harming the growth of the organism. Now let's know about nitrogen sources. Cause drastic reduction in citric acid production without harming the growth of the organism. Now let's know about nitrogen sources. Ammonium salts, nitrates and uric acids are the nitrogen sources used in the media for the citric acid production. All the three compounds are equally good sources as long as they do not adversely affect pH of the medium. Okay, next is oxygen supply cause drastic reduction in citric acid production without harming the growth of the organism. Now let's know about nitrogen sources. Ammonium salts, nitrates and urea are the nitrogen sources used in the media for citric acid production. All the three compounds are equally good sources as long as they do not adversely affect the pH of the medium. If molasses are used for nutrient supply, addition of extra nitrogen sources is required. However, some workers have shown that exogenous addition of ammonium ions stimulates citric acid production. Now let's know about production processes of citric acid. There are two processes by which citric acid can be industrially produced. First is the surface process, second is the submerged process. Let's know about first process, the surface process. This is characterized by growing the microorganism as a layer or a film on a surface in contact with a nutrient medium, which may be solid or liquid in nature. Thus, the surface process has supported growth systems. Now, the second process is, let's know about the second process, that is the submerged process. In this case, the organisms are immersed or dispersed throughout the nutrient medium. There are two types of submerged fermentation, one stirred bioreactor and the airlift bioreactor. So let's know about the surface process. Sit here. No. Let's know about surface processes. First is solid surface fermentation. Solid process using solid substrates are particularly carried out in less developed areas of some Asian countries. The solid substrates such as wheat bran or pulp from sweet potato starch are used as culture media. The pH of the medium is used as the pH of the medium is adjusted to 4 to 5 and then sterilized. Now the inoculum in the form of spores of A. niger is spread as layer 3 to 6 cm thickness and incubated at 28 degrees centigrade. The growth of the organism can be accelerated by the addition of alpha amylase. The solid state fermentation takes about 80 to 100 hours for maximal production of acetic acid. At the end of the process, citric acid can be extracted into hot water and isolated. Now let's know about liquid surface fermentation. Surface fermentation using liquid as a nutrient medium is the oldest method for citric acid production. It is still in use due to the simple technology, low energy cost and higher reproducibility. Further, the interference of trace metals and dissolved oxygen tension are minimal. The labor costs are however higher since the manpower and requirements are more for cleaning the systems. About 20% of the citric acid in the world is produced by surface processes. The nutrient supply for surface fermentation normally comes from the beet molasses. The fermentation is usually carried out in aluminum trays filled with sterile nutrient medium. The inoculum in the form of spores is spread over the medium. A sterile air is passed for supplying oxygen as well as cooling. The temperature is maintained around 30 degrees centigrade during fermentation. As the spores germinate, that occurs within 24 hours of inoculation, a layer of mycelium is formed over the medium. The pH of the nutrient medium falls to less than 2 as the mycelium grows in size and forms a thick layer on the surface of the nutrient solution. The fermentation is stopped after 7 to 15 days. Now let's know about submerged processes. Around 80% of the world's supply of citric acid is produced by submerged processes. This is the most preferred method due to its high efficiency and easy automation. The disadvantages of the submerged fermentation are adverse influence of trace metals and other impurities, variations in oxygen tension and advanced control technology that requires highly trained personnel. Two types of bioreactors are in use, tiered tanks and aerated towers. The vessels of the bioreactors are made up of high quality stainless steel. The spurging of air occurs from the base of the fermenter. The success and the yield of citric acid production mainly depends on the structure of mycelium. The mycelium with forked and bulbous hypha and branches with aggregate into pellets is ideal for citric acid formation. On the other hand, no citric acid production occurs if the mycelium is loose and fermentation with limited branches. An adequate supply of oxygen is required for good production of acetic acid of citric acid. The ideal aeration rate. Now let's know about production of citric acid from alkanes. Both yeast and bacteria can be used for citric acid production from N-alkanes, that is C9 to C means carbon 9 to carbon 23 hydrocarbons. The citric acid yield is better from hydrocarbons compared to sugars. That is 145% of hydrocarbons compared to sugars, that is 145% of the citric acid from paraffin. The most commonly used organism is Candida lipolytica. The fermentation can be carried out in batch, semi-continuous or continuous modes. The pH should be kept above 5. The major limitations of citric acid production from alkanes are very low solubility of alkanes and increased production of unwanted isocitric acid. Now let's know about recovery of citric acid. The steps for the recovery of citric acid either from surface process or submerged process are comparable. The recovery starts with the filtration of culture broth and washing of mycelium. Oxalic acid is an unwanted byproduct and it can be removed by precipitation by adding lime at pH below 3. The culture broth is then subjected to pH 7.2 and temperature 70 to 90 degree for precipitating citric acid. For further purification, citric acid is dissolved in sulfuric acid. The final steps for citric acid recovery are treatment with activated charcoal, cation and anion exchangers and crystallization. Citric acid monohydrate formed below 36 degrees centigrade is the main commercial product. Above 40 degrees centigrade, citric acid crystallizes in an anhydrous form. The degree of purity of citric acid produced depends on the purpose for which it is required. For instance, pure forms of citric acid are needed for use in food preparations, while for industrial use it can be crude form. Now let's know about gluconic acid. Gluconic acid can be produced by several bacteria and fungi. Glucose on a simple direct dehydrogenation forms a D-gluconolactone which is then converted to gluconic acid. Now let's know about applications of gluconic acid. First point is gluconic acid is used in the manufacture of metals, stainless steel and leather as it can remove the calcareous and rust deposits. Second is it is used as an additive to foods and beverages. Third is gluconic acid has pharmaceutical applications, calcium and iron therapy. Fourth is sodium gluconate is used as a sequestering agent in many detergents. Fifth is gluconate is used for de-sizing polyester or polyamide fabrics. Sixth is it is utilized in the manufacture of highly resistant concrete. Now let's know about microbial production of gluconic acid. Gluconic acid can be produced by a wide variety of prokaryotic and eukaryotic microorganisms. Bacterial species of the genera, that is gluconobacteria, gluconobacter, Acetobacter, Pseudomonas vibrio. Now let's know about fungal species of genera. That are Aspergillus, Penicillium, Glyocladium. Let's know about the principle of production. The enzymatic reaction for the formation of gluconic acid in gluconobacter suboxidants and Aspergillus niger are we will discuss. In bacteria, intracellular glucose is converted to extracellular gluconic acid. A membrane-bound enzyme, glucose dehydrogenase, utilizes quinone, that is in short PQQ, as coenzyme and converts glucose to delta D gluconol acetone, which undergoes hydrolysis to form gluconic acid. As regards fungal production, glucose is oxidized by the extracellular enzyme. Glucose oxidase is to form the delta D gluconol acetone, which subsequently gets converted to gluconic acid by lactonase. Glucose oxidase is an inducible enzyme that can be induced by high concentration of glucose and at pH above 4. It is believed that H2O2 produced by glucose oxidase acts as an antagonist against other microorganisms in the surroundings. Now let's know about production process for gluconic acid. Submerged processes by employing either A niger or G suboxidants are used for producing gluconic acid. The culture medium contains glucose at a concentration of 12 to 15%. The fermentation is carried out at pH 4.5 to 6.5 and at temperature 28 to 30 degrees centigrade for a period of about 24 hours. Increasing the supply of oxygen enhances gluconic acid yield. Biotechnologists exploit the fermentation process of gluconic acid for the production of the enzyme glucose oxidase besides producing calcium gluconate and sodium gluconate. Chemical synthesis. Let's know about the chemical synthesis of gluconic acid. By employing the immobilized enzyme glucose oxidase, gluconic acid can also be produced. Now let's know about lactic acid. Lactic acid occurs in two isomeric forms, that is L plus and D minus isomers, and as a racemic mixture, that means DL lactic acid. The isolation of lactic acid from milk was done in 1798. It was the first organic acid produced by microorganisms in 1880. Today, lactic acid is comparatively produced both by microbiological and chemical methods. Applications of lactic acid. Let's know about the applications of lactic acid. There are different grades of lactic acid, mainly besides on the percentage of lactic acid, the grades and their applications. Now we will discuss microorganisms for the production. Let's know about the microorganisms for the production of lactic acid. Lactic acid producing bacteria are broadly categorized into two types: heteroformative bacteria and homoformative bacteria. Now first let's know about heteroformative bacteria. It produces other byproducts besides lactic acid and therefore are not useful for industrial production of lactic acid. These bacteria are employed in food or feed preservation. Now let's know about homoformative bacteria, specialized for exclusive production of lactic acid and therefore are suitable for the industrial purposes. Now let's know about lactobacillus species. Lactobacillus species are used for lactic acid production. However, there are variations in the substrate utilized, and let's know about this. L. delbrueckii and L. leishmani is here glucose. L. bulgaricus, L. helvetii. It is found in whey. There is lactose, presence of lactose. L. lactis, it is from maltose. L. amylophilus, that is starch. L. pentosus, that is from sulfite waste liquor. Now let's know about the biosynthesis of lactic acid. The synthesis of lactic acid occurs through glucose oxidation by glycolysis to produce pyruvate, which on reduction gives lactic acid. The reducing equivalents produced during the oxidation of glyceraldehyde 3-phosphate are utilized by the enzyme lactate dehydrogenase to form lactate. Most of the lactic acid producing microorganisms normally produce only one isomer of lactic acid, L plus or D minus. However, some bacteria which usually occur as infection can form racemic mixture. Now let's know about production process for lactic acid. The fermentation medium contains 12 to 15% of glucose, nitrogen and phosphate containing salts and micronutrients. The process is carried out at pH 5.5 to 6.5 and temperature 45 to 50 degrees centigrade for about 75 hours. Generally, the strains operating at higher temperature, that means 45 to 60 degrees centigrade, are preferred since it reduces the need for medium sterilization. As the lactic acid is produced, it has to be removed since it is toxic to the organism. This can be achieved either by a continuous culture technique or by removal of lactic acid by electrodialysis. Theoretically, every molecule of glucose forms two molecules of lactic acid. About 90% of theoretical yields is possible in fermentation industry. L plus lactic acid is predominantly produced. The outline of the steps involved in the lactic acid we will discuss. Now let's start. Let's know about acetic acid. The production of acetic acid in the form of vinegar, that is used as a refreshing drink from alcoholic liquids, has been known for centuries. Now let's know about the microorganisms used for the production of acetic acid. The commercial production of acetic acid is carried out by a special group of acetic acid bacteria which are divided into two genera. Gluconobacter. Let's know about gluconobacter, and second is acetobacter. Gluconobacter that oxidizes ethanol exclusively to acetic acid, and acetobacter that oxidizes ethanol first to acetic acid and then to carbon dioxide and water. These over-oxidizes are gram negative and acid tolerant. Example A acetic, A peroxidans, A pasturinianus. Now let's know about the biosynthesis of acetic acid. Acetic acid is a product of incomplete oxidation of ethanol. Ethanol is first oxidized by alcohol dehydrogenase to acetaldehyde, which then gets dehydrated to form acetaldehyde hydrate. The lattice is then acted upon by acetaldehyde dehydrogenase to form acetic acid. Now let's know about the production process for acetic acid. For every molecule of ethanol oxidized, one molecule of acetic acid is produced. Thus, high-yielding strains can produce 11 to 12% acetic acid from 12% alcohol. For optimal production, adequate supply of oxygen is very essential. Inefficient oxygen coupled with high concentration of alcohol and acetic acid result in the death of microorganisms. Surface fermentation or submerged fermentation process can be carried out to produce acetic acid. Trickling generation process, a type of surface fermentation, is very commonly used. Now let's know about the recovery. The acetic acid produced in is clarified by filtration and then subjected to decolorization by K4 FeCN whole 6. Now let's know about the production of vinegar. Vinegar is an aqueous solution containing about 4% by volume acetic acid and small quantities of alcohol, salts, sugars and esters. It is widely used as a flavoring agent for processed liquid foods such as sauce and ketchup. The starting material for vinegar production are wine, whey, malt, and vinegar production can be carried out either by surface process or by submerged process. Let's know about the surface process. What do we mean by surface process? The fermentation material is sprayed over the surface which trickles through the shavings that contain the acetic acid producing bacteria. The temperature is around 30 degrees centigrade on the upper part while it is around 35 degrees centigrade on the lower part. The vinegar is produced in about 3 days. Now let's know about submerged process. The fermentation bioreactors are made up of stainless steel. Aeration is done by a suction pump from the top. The production rate in the submerged process is about 10 times higher than the surface process. Now let's know about L-ascorbic acid. L-ascorbic acid is a commonly used chemical name for the water soluble vitamin C. This vitamin forms a redox system and participates in several biological processes. It is intimately involved in the biosynthesis of collagen, the most abundant protein in human body. It is the most abundant protein in the human body. Vitamin C also protects the body against carcinogenic nitrosamines and free radicals. The deficiency of ascorbic acid causes scurvy. Now let's know about the applications of ascorbic acid. Because of the wide range of physiological and beneficial functions of ascorbic acid, its commercial production assumes significance. Vitamin C is mainly used for food and pharmaceutical industries. Now let's know about the industrial production of ascorbic acid. Ascorbic acid is commercially produced by a combination of several chemical steps and one reaction of biotransformation brought out by microorganisms. This process is referred to as Risten-Grussner synthesis. D-glucose is first converted to d-sorbitol. Oxidation of d-sorbitol to L-sorbose is carried out by Acetobacter xylenum or A suboxidants. A submerged bioreactor fermentation process is ideal for this reaction. It takes about 24 hours at temperature 30 to 35 degrees centigrade. Sorbose by a couple of chemical reactions can be finally converted to L-ascorbic acid. Normally, about 100 gram of ascorbic acid is produced from 200 gram of glucose in Risten-Grussner synthesis. Now, let's know about two-step fermentation process. In this, D-glucose is converted to 2,5-diketogluconic acid by Erwinia Acetobacter or Gluconobacter species. In the second step, Corynebacterium species converts 2,5-diketogluconic acid or 2,2-keto-L-gluconic acid. It is also possible to involve Bacillus megaterium for converting L-sorbose to 2-keto-L-gluconic acid. The latter by chemical reactions can be converted to ascorbic acid. Now let's know about the production via L-gulonol acetone. Ascorbic acid can also be synthesized via gulonol acetone, which can be directly converted to L-ascorbic acid by the enzyme L-gulonol acetone dehydrogenase. Now let's know about the direct production of ascorbic acid by fermentation. Several workers are trying to produce ascorbic acid directly from glucose. Microalgae of Chlorella have also have shown some promising results, although the yield is very low. Now let's know about genetic engineering for ascorbic acid production. Biotechnologists have been successful in cloning and expressing the gene for 2,5-diketogluconic acid reductase of Corynebacterium species into Erwinia horticola. By cloning this, the two-step fermentation process has been reduced to one. The genetically engineered Erwinia cells were able to convert D-glucose directly to 2-keto-L-gluconic acid. This is certainly advantageous since the metabolic capabilities of two different microorganisms could be combined into one organism. However, the yield of ascorbic acid by the hybrid strain was very low. Scientists are now trying to alter certain amino acids into 5-diketogluconic acid reductase and increase the catalytic activity of this enzyme. Now let's know about the itaconic acid. Itaconic acid is used in plastic industry, paper industry and in the manufacture of adhesives. Itaconic acid can be commercially produced by Aspergillus itoconicus and A. terras. The biosynthesis of itaconic acid occurs by way of Krebs cycle. The metabolite cis-aconic acid undergoes decarboxylation catalyzed by the enzyme cis-aconitic decarboxylase. Itaconic acid is oxidized to tartaric acid by itaconic acid oxidase. This enzyme has to be inhibited for a maximum yield of itaconic acid. This can be achieved by adding calcium. Batch submerged fermentation process is commonly used for itaconic acid production. The yield is around 75% of the theoretical calculation when the medium contains 15% sucrose. Now, finally completed. Any questions regarding this, you can ask me in the comment box and I'll definitely answer the question. And please kindly subscribe my channel for getting more updates. Thank you.